So coming back to this, you should have that the A has one really short one, one longer one, and one kind of in between. All right, so the really short one's going to be way over here. All right, it's going to travel pretty far. So this is number 1. And then, so this one goes the farthest. The number 3 will be closer to the well, because it's going to be bigger. And then 2 is not as quite as small as here, but closer to the 3, so maybe about right here. All right. So then in the B range here, they also have 1. The 1 is a little bit bigger, don't you think? Than this one, but this one is even smaller, number 3. So the 3 is going to actually travel even further than the 1 over here. So that's going to travel the furthest. All right, then this one is a little bit bigger than this one here. So therefore, but not quite as big as the 2. All right, so therefore this is not going to be over here with this 2 right here, but not quite as far as this. So looking at this, it's about twice as big as this 1, but about as half as big as this 2. So it's probably going in this row somewhere in here. So you're 2, and then, or sorry, 1. That's 1, not 2. All right, 1. And then the 2 is really big, so that's going to be really over here. So this is going to be closer to the well here. So this is 2. So this is supposed to be all one row. This is supposed to be all one lane. And then C, this one looks exactly like this one here. So that's going to be about right in the same area here. So that's 1. All right, and then look at these are about in the same spot. So the 2s. And so if this was a crime scene, and then the 3, if that's the same, 2 and 3, then the crime scene DNA seems to match suspect 2. All right, and so that's how we look. And they call these a DNA fingerprint because it is unique to individuals, and so just like fingerprints are. And so you look at that banding pattern, and we can say, oh, suspects 2 DNA is found at the crime scene. It doesn't mean that suspect 2 is guilty. It just means that they're found at the crime scene and can be used as evidence against that particular person. So that's another use of gel electrophoresis. So if we flip over here, restriction fragment length polymorphisms are called RFLPs for short. These are differences in DNA sequences on homologous chromosomes that result in restriction fragments of different lengths. So we saw this with the sickle cell gene. We have homologous chromosomes, and we have differences between them, causing them to cut different, and therefore end up in different lengths of fragments. And so this RFLP can serve as genetic markers for a particular location in the genome. Okay? And so for concept 20.3, it says entire genomes can be mapped at the DNA level. So we have mapped the whole human genome and figuring out the A's, T's, C's, and G's. So the most ambitious mapping project to date has been the sequence of the human genome. Officially begun as the Human Genome Project in 1990, the sequencing was completed. Most of it was completed by 2003. And so we have sequenced, so what do we mean by that is the order of A's, T's, C's, and G's, etc. in the whole human genome. And we've also done that with other organisms as well, so other genomes, and so therefore allows us to compare DNA between humans and other organisms and one organism to another. All right, and this is where I'm going to stop in the video for tonight.